Hello students, today we'll be dealing with the bioenergetics of metabolic cycle part 1, which is glycolysis. Let's start with the introduction first. Glucose occupies a central position in the metabolism of plants, animals and many microorganisms. It is relatively rich in potential energy and thus a good fuel. By storing glucose as a high molecular weight polymer, such as starch or glycogen, a cell can stockpile large quantities of hexose units while maintaining a relatively low cytosolic osmolarity. When energy demands increase, glucose can be released from these intracellular storage polymers and used to produce ATP either aerobically or anaerobically. Glycolysis is an almost universal central pathway of glucose catabolism, the pathway with the largest flux of carbon in most cells. The glycolytic breakdown of glucose is the sole source of metabolic energy in some mammalian tissues and cell types like the erythrocytes, the renal medulla, brain, and sperm, for example. Some plant tissues that are modified to store starch, such as potato tubers, and some aquatic plants like watercress derive most of their energy from glycolysis. Many anaerobic microorganisms are entirely dependent on glycolysis. Glycolysis, also known as the Embden-Meyerhof pathway, is a sequence of reactions that metabolizes one molecule of glucose to two molecules of pyruvate with the concomitant net production of two molecules of ATP. This process is anaerobic, that is, it does not require molecular oxygen. Pyruvate can be further processed anaerobically to lactate in the case of lactic acid fermentation or ethanol in the case of alcoholic fermentation. Under aerobic conditions, pyruvate can be completely oxidized to carbon dioxide, generating much more ATP. In eukaryotic cells, glycolysis takes place in the cytoplasm. This pathway consists of three stages. Stage 1, which is the conversion of glucose into fructose 1,6-biphosphate. It consists of three steps: a phosphorylation, an isomerization, and a second phosphorylation reaction. The strategy of these initial steps in glycolysis is to trap the glucose in the cell and form a compound that can be readily cleaved into phosphorylated 3-carbon units. Stage 2 is the cleavage of the fructose 1,6-biphosphate into two 3-carbon fragments. These resulting 3-carbon units are readily interconvertible. In stage 3, ATP is harvested when the 3-carbon fragments are oxidized to pyruvate. Let's look at the preparatory phase of glycolysis. In the preparatory phase of glycolysis, two molecules of ATP are invested and the hexose chain is cleaved into 2-triose phosphate. Number 1. Phosphorylation of glucose. In the first step of glycolysis, glucose is activated for subsequent reactions by its phosphorylation at C6 to yield glucose 6-phosphate with ATP as a phosphoryl donor. This reaction, which is irreversible under intracellular conditions, is catalyzed by hexokinase. Kinases are enzymes that catalyze the transfer of the terminal phosphoryl group from ATP to an acceptor nucleophile. Hexokinase, like many other kinases, requires magnesium ions for its activity because the true substrate of the enzyme is not ATP but the magnesium ATP complex. Next, conversion of glucose 6-phosphate to fructose 6-phosphate. The enzyme phosphohexose isomerase or phosphoglucose isomerase catalyzes the reversible isomerization of glucose 6-phosphate, an aldose, to fructose 6-phosphate, which is a ketose. The reaction proceeds readily in either direction. This isomerization has a critical role in the overall chemistry of the glycolytic pathway, as the arrangement of the carbonyl and the hydroxyl groups at C1 and C2 is a necessary prelude to the next two steps. Now let's look at the phosphorylation of fructose 6-phosphate to fructose 1,6 biphosphate. In this step, phosphofructokinase 1, also known as PFK1, catalyzes the transfer of a phosphoryl group from ATP to fructose 6-phosphate to yield fructose 1,6 biphosphate. The PFK1 reaction is essentially irreversible under cellular conditions, and it is the first committed step in the glycolytic pathway. Glucose 6-phosphate and fructose 6-phosphate have other possible fates, but fructose 1,6 biphosphate is targeted for glycolysis. The activity of PFK1 is increased whenever the cell's ATP supply is depleted, or when the ATP breakdown products, ATP and AMP, particularly the latter one, are in excess. The enzyme is inhibited whenever the cell has ample ATP and is well supplied by other fuels such as fatty acids. Now let's look at the next step, which is cleavage of fructose 1,6 biphosphate. The enzyme fructose 1,6 biphosphate aldolase, often called simply aldolase, catalyzes a reversible aldol condensation. Here, fructose 1,6 biphosphate is cleaved to yield two different triose phosphates, glyceraldehyde 3-phosphate, which is an aldose, and dihydroxyacetone phosphate, which is a ketose. Now the next step is interconversion of the triose phosphates. Only one of the two triose phosphates formed by the aldolase, which is glyceraldehyde 3-phosphate, can be directly degraded in the subsequent steps of glycolysis. The other product, which is dihydroxyacetone phosphate, is rapidly and reversibly converted to glyceraldehyde 3-phosphate by the fifth enzyme of the sequence, which is triose phosphate isomerase. This reaction completes the preparatory phase of glycolysis. The hexose molecule has been phosphorylated at C1 and C6 and then cleaved to form two molecules of glyceraldehyde 3-phosphate. Now let's look at the payoff phase of glycolysis. The payoff phase of glycolysis includes the energy-conserving phosphorylation steps in which some of the free energy of the glucose molecule is conserved in the form of ATP. We know now that one molecule of glucose yields two molecules of glyceraldehyde 3-phosphate. Both halves of the glucose molecule follow the same pathway in the second phase of glycolysis. The conversion of two molecules of glyceraldehyde 3-phosphate to two molecules of pyruvate is accompanied by the formation of 4 molecules of ATP from ADP. However, the net yield of ATP per molecule of glucose degraded is only 2 because the 2 ATP were invested in the preparatory phase of glycolysis to phosphorylate the 2 ends of the hexose molecule. Now let's look at the sixth step, which is oxidation of glyceraldehyde 3-phosphate to 1,3 biphosphoglycerate. The first step in the payoff phase is the oxidation of glyceraldehyde 3-phosphate to 1,3 biphosphoglycerate, catalyzed by glyceraldehyde 3-phosphate dehydrogenase. This is the first of the 2 energy-conserving reactions of glycolysis that eventually lead to the formation of ATP. The aldehyde group of glyceraldehyde 3-phosphate is oxidized, not to a 3-carboxyl group, but to a carboxylic acid anhydride with phosphoric acid. This type of anhydride, called an acyl phosphate, has a very high standard free energy of hydrolysis. Much of the free energy of oxidation of the aldehyde group of glyceraldehyde 3-phosphate is conserved by formation of the acyl phosphate group at C1 of 1,3 biphosphoglycerate. Let's look at the 7th step, which is phosphoryl transfer from 1,3 biphosphoglycerate to ADP. The enzyme phosphoglycerate kinase transfers the high-energy phosphoryl group from the carboxyl group of 1,3 biphosphoglycerate to ADP, forming ATP and 3-phosphoglycerate. Steps 6 and 7 of glycolysis together constitute an energy coupling process in which 1,3-biphosphoglycerate is the common intermediate. It is formed in the first reaction, which would be endergonic in isolation, and its acyl phosphate group is transferred to ADP in the second reaction, which is strongly exergonic. The formation of ATP by phosphoryl group transfer from a substrate such as 1,3-biphosphoglycerate is referred to as a substrate-level phosphorylation. Substrate-level phosphorylations involve soluble enzymes and chemical intermediates, 1,3-biphosphoglycerate in this case. Now let's look at step 8, which is conversion of 3-phosphoglycerate to 2-phosphoglycerate. The enzyme phosphoglycerate mutase catalyzes a reversible shift of the phosphoryl group between C2 and C3 of the glycerate. Magnesium ion is essential for this reaction. Now, step 9, dehydration of 2-phosphoglycerate to phosphoenol pyruvate. In the second glycolytic reaction that generates a compound with high phosphoryl group transfer potential, enolase promotes reversible removal of a molecule of water from 2-phosphoglycerate to yield phosphoenol pyruvate, also known as PEP. Step 10, which is transfer of the phosphoryl group from phosphoenol pyruvate to ADP. The last step in glycolysis is the transfer of the phosphoryl group from phosphoenol pyruvate to ADP, which is catalyzed by pyruvate kinase and requires potassium ions and either magnesium or manganese ions. In the substrate-level phosphorylation, the product pyruvate first appears in its enol form, then tautomerizes rapidly and non-enzymatically to its keto form, which predominates at pH 7. The overall reaction has a large negative standard free energy change due in large part to the spontaneous conversion of the enol form of pyruvate to the keto form constitutes a large driving force, pushing the reaction towards ATP synthesis. The pyruvate kinase reaction is essentially irreversible under intracellular conditions and is an important site of regulation. So, in the overall glycolytic process, one molecule of glucose is converted to 2 molecules of pyruvate, and this is the pathway of carbon. 2 molecules of ADP and 2 of inorganic phosphate are converted to 2 molecules of ATP, and this is the pathway of the phosphoryl groups. 4 electrons as 2 hydride ions are transferred from 2 molecules of glyceraldehyde 3-phosphate to 2 of NAD+, and this is the pathway of the electrons. The net equation for the overall process is C6H12O6 plus 2 NAD plus 2 ADP plus 2P, which gives 2 pyruvic acid plus 2 ATP plus 2 NADH plus 2 hydrogen ions. Now let's look at the regulation of glycolysis. During his studies on the fermentation of glucose by yeast, Louis Pasteur discovered that both the rate and the total amount of glucose consumption were many times greater under anaerobic than aerobic conditions. This is known as Pasteur effect, according to which the introduction of oxygen brings about a decrease in the rate of glycolysis. The ATP yield from glycolysis under anaerobic conditions, that is 2 ATP per molecule of glucose, is much smaller than that from the complete oxidation of glucose to carbon dioxide under aerobic conditions, which is about 30 or 32 ATP per glucose. About 15 times as much glucose must therefore be consumed anaerobically to yield the same amount of ATP as aerobically. The flux of glucose through the glycolytic pathway is regulated to maintain nearly constant ATP levels. The required adjustment in the rate of glycolysis is achieved by a complex interplay among ATP consumption, NADH regeneration, and allosteric regulation of several glycolytic enzymes, including hexokinase, PFK1, and pyruvate kinase, and by second-to-second fluctuations in the concentration of key metabolites that reflect the cellular balance between ATP production and consumption. On a slightly longer time scale, glycolysis is regulated by the hormones glucagon, epinephrine, and insulin, and by changes in the expression of the genes for several glycolytic enzymes. Now let's come to the conclusion. Glycolysis is a near universal pathway by which a glucose molecule is oxidized to two molecules of pyruvate with energy conserved as ATP and NADH. All 10 glycolytic enzymes are in the cytosol, and all 10 intermediates are phosphorylated compounds of 3 or 6 carbons. In the preparatory phase of glycolysis, ATP is invested to convert glucose to fructose 1,6-biphosphate. The bond between C3 and C4 is then broken to yield 2 molecules of glyceraldehyde 3-phosphate. In the payoff phase, each of the 2 molecules of glyceraldehyde 3-phosphate derived from glucose undergoes oxidation at C1. The energy of this oxidation reaction is conserved in the formation of 1 NADH and 2 ATP per triose phosphate oxidized. The net equation for the overall process is 1 molecule of glucose plus 2 NAD plus 2 ADP plus 2P gives 2 molecules of pyruvic acid plus 2 ATP plus 2 NADH plus 2 hydrogen ions. Glycolysis is tightly regulated in coordination with other energy-yielding pathways to assure a steady supply of ATP. Hexokinase, PFK1, and pyruvate kinase are all subject to allosteric regulation that controls the flow of carbon through the pathway and maintains constant levels of metabolic intermediates.